Hello everyone. In this chapter 3, we will cover meiosis, the development, and aging. Because of the length of the chapter, however, we are going to divide the video presentation in two parts. The first part is going to focus on reproductive organs and meiosis, while the second part will focus on the development and aging. Now, what are some learning outcomes of this chapter? In the first aspect, we expect you to know is to understand the different parts of the reproductive systems, both in male and females, the process of meiosis, which is a process by which gametes are produced. Then we are going to ask the following question: what are the different steps of gamete production, both in males and in females? Then we are going to dive in the prenatal development. We're going to address birth defects. And finally, we hope to learn about maturation and aging. These are the 6 learning outcomes for this chapter. Without further ado, let's take a look at the reproductive systems. Now, as we consider the human body from eggs to full development, genes orchestrate our physiology right after fertilization. Fertilization being the time when the sperm cell meets the egg. From that point, we have an embryo that forms, that we have a zygote first, then an embryo, and from the embryo, we're going to have a fetus, and from a fetus, we're going to have a baby to full growth. Genes are going to determine the different changes that are going to happen through the development process. Now, in reproduction, obviously, the first step is going to be the production of sex cells. These sex cells are the sperm in males and eggs or oocytes in females. Now, the first cell is going to form when a sperm meets an oocyte or an egg, right? So this is going to form a zygote. Now, the sperm cells and eggs are also called gametes or sex cells. Sex cells or reproductive cells. So, these cells are produced in gonads. Gonads exist by pair in both female and males, right? For instance, the testicles in males are going to be the gonads, right? You have 2 testicles. The ovaries, you're going to have 2 ovaries in female, while testicles are going to be in male. Then, after production of the gametes, you're going to have some tubular structures that are going to transport these gametes to the site of fertilization. In addition, we're going to have hormones and secretions that are going to play a very important role in controlling reproduction. Without these hormones and this secretion, it would be very difficult to reach an adult individual able to reproduce. So, let's first make a stop in the male reproductive organs / male reproductive system. Now, we are going to have the seminiferous tubes. These seminiferous tubes are the tubes where the stem cells are going to differentiate to become sperm cells. These seminiferous tubes are going to be found in testes. This is where the sperms are going to be produced. Once the sperms are produced, they're going to be stored and mature in the epididymis. Epididymis. Okay? The testes are carried by a sac that is called a scrotum. Now, going back to the epididymis, the epididymis is going to fold, and then we have a tube that is going to develop from the epididymis going up this way. That tube is the duct deferens. The duct deferens is a tube through which the sperm cells are going to travel during ejaculation. From the epididymis, they're going to be carried to the duct deferens. From there, the sperm is going to fall in this tube right here. Now, this tube right here, it's an area where a fluid that is called the seminal fluid is going to be released. Seminal fluid. Now, that seminal fluid is released to prepare the way for sperms to go through the urethra without being destroyed. Now, the seminal fluid is produced by a set of 3 organs. We're going to have the seminal vesicles, the prostate gland, and the bulbourethral gland. These 3 structures are going to produce the seminal fluid, which again is going to prepare the urethra as sperms go through the urethra and released outside via the penis. So it is very important to remember the fact that the seminal fluid is going to be essential to keep the integrity of this sperm cells. Now, the penis is going to be essential for transporting the sperm cells to the vagina, and eventually the sperm cells are going to swim and be able to go through the uterus and through the fallopian tubes, where hopefully at some point they're going to encounter an egg. But let's close this parenthesis for now. And let's mention that the scrotum that covers the testes is low in the body and relatively cold. This is essential, relatively cold compared to the remainder of the body, obviously. This is essential because sperm cells are produced and mature better in an environment that is a little colder compared to the inside of the body, like in the muscles. So, I did tell you about the the urethra, okay, which is the canal through which the sperm is going to leave the reproductive system. Then, as part of the system, you're also going to have the urinary bladder. But really, the urinary bladder doesn't play a role in reproduction, so let's move on here. So the sperm cells are made in the seminiferous tubules. The seminiferous tubules are found in the testes. The sperm cells are going to mature and be stored in the epididymis until ejaculation. The prostate gland, the seminal vesicles, and the bulbourethral gland secrete the seminal fluid. I mentioned that. And the sperm from each duct deferens is going to go through the penis and eventually go in the vagina. How about the reproductive organs in a female? So we're going to have some fleshy external part of the reproductive organs that are going to be the labia major and the labia minor. Now, beyond the labia minor and the labia major, we're going to have the vagina. Okay? Then, beyond the vagina, we're going to have the cervix. Then, we're going to have the fallopian tubes or the uterine tube and the ovaries. The ovaries are the organs that produce oocytes or egg cells. Once these egg cells are produced, they are going to travel through the fallopian tube or the uterine tube. Now, let me say that while a male is making sperm cells from the age of puberty to adulthood, a female will have a specific batch of egg cells that which formation starts in the prenatal stage, and then before birth, this batch of cells is going to stop further cell division. They're going to stop at very specific stage of meiosis. And then when a lady reaches puberty, the age of puberty, then every month an egg is going to be released in the fallopian tubes by the ovaries, and that egg is going to travel along the tube. And if that egg is encountered by a sperm cell, that egg is going to be fertilized and further development is going to happen. We are going to address that a little later. If that egg is not fertilized, what will happen is that the egg will keep migrating toward the uterus, and part of the uterus is going to be released along with the wasted egg in a cycle of menstruation, right? So we're going to see loss of blood in that situation. So also in terms of reproduction, you have an organ here, the clitoris, which is about 2 centimeters long. That clitoris actually plays the role of the penis in a female. That clitoris can be actually stimulated, and this is going to allow a lady to reach orgasm, which is actually essential for muscle contraction to occur to actually push the egg toward the uterus. All right, so the oocytes or egg cells mature in the ovaries, and each month an ovary is going to release an egg. And that egg is going to travel through the uterine tube. And as I mentioned, if the egg is fertilized, it will continue to move toward the uterus and divide at the same time and develop. If the egg is not fertilized, that egg is going to be expelled, right? Along with some part of the lining of the uterus in the menstrual flow. Hormones are going to control the cycle of the oocyte maturation and the preparation of the uterus to nurture the fertilized egg, right? And that fertilized egg, at that point, we don't call it egg anymore. We call it a zygote. And that zygote is going to multiply to form a mass of cell, and development is going to occur from there. Now, let's take a look at meiosis. What is meiosis? Meiosis, it's a type of cell division. That type of cell division is unique in the sense that that cell division is going to produce genetic diversity. That's one. Two, it's a reduction division. In other words, we go from a cell that has 46 chromosomes, so 23 pairs of chromosomes, to a cell that has 23 chromosomes. We go from a cell that is haploid to cells that are haploid. We're going from cells that are diploid to cells that are haploid. Anyway, let me go back. So meiosis is a type of cell division. That type of cell division is going to divide in half the amount of chromosomes at the end of the day in each daughter cells. Now, specifically, homologous chromosomes. You receive a set of chromosome from mom and a set of chromosome from dad, right? And each chromosome you receive from mom has its equivalent in dad. We call them homologous chromosomes, right? Homologous pairs are going to have exactly the same genes, even though these genes may differ in terms of the sequence of nucleotide. We talked about alternate forms of the same gene, right? So we are going to have the genes at the very same location on the chromosome. However, the fact that there may be variant form of the same gene is going to cause us to call them alleles, right? So variant alleles of the same genes. The gametes are going to be haploid cells. They contain half the amount of chromosomes that you would find in a somatic cell that are in a somatic cell that is a diploid cell, right? Absence of meiosis can cause too much chromosomes in an individual. So it is not typical that an individual is going to have a duplication of the genetic information. Anytime there is too much DNA, we're going to see a spontaneous miscarriage that is going to happen. Meiosis, it's a mix-up thread recombination. In other words, you have a mingling of the genetic information so that there is hardly any chance, if any chance at all, that two gametes are the same. That doesn't occur, right? Meiosis provides a huge diversity in the genetic array that we see in a human population, right? Even within a family, we see a genetic diversity because meiosis makes it possible. There are some mechanisms by which meiosis is making that genetic diversity possible. One, it's the law of independent assortment. Two, it's the crossing overs. We are going to address that when we look at the details of meiosis. Meiosis consists of two divisions. In the first division that we call meiosis 1, we're going to have that the two homologous chromosomes are going to end up into two separate cells. That causes us to call meiosis 1 a reduction division. Why? Because we are going from pairs of homologous chromosomes to one single unit, right? So, for instance, you have this as the chromosome you receive from mom, and this the chromosome you receive from dad. After meiosis 1, this one end up in one cell, and this one end up in another cell. Now they are in two separate cells, right? This is a reduction division. Now, prior to that division though, there is an interphase, like in a normal cell, right? Now, after that meiosis 1, we have a second division. That second division, meiosis 2, follows quickly after meiosis 1, and between meiosis 1 and meiosis 2, there is no replication of DNA. Because of that, we call this division, meiosis 2, equational division. Because at this point, the chromosome that you receive from dad, now which are in the same cell, are going to further divide. Now, the sister chromatids of the chromosome you receive from mom are going to divide, and the sister chromatid you receive from dad are also going to divide, right? But at the end of the day, we have 23 chromosomes in the gamete at the end of the day. So, meiosis 1 is going to, meiosis is going to be preceded by interphase. I already said that. Each division is going to have a prophase, a metaphase, an anaphase, and a telophase / cytokinesis. Now, how do mitosis compares with meiosis? During mitosis, we have one division, and that one division is going to cause the development of two daughter cells. These two daughter cells are going to be genetically identical. On the other hand, though, meiosis is a set of two consecutive divisions. These two consecutive divisions are going to produce four daughter cells. These four daughter cells are going to be genetically different. It is very important to remember this comparative details. Then the second aspect I would like to focus on the meiosis side, the cell goes from 2N chromosome to producing daughter cells that have 2N chromosome as well. So the cell we start with was diploid. The daughter cells are diploid. However, in meiosis, the parent cell, while it is diploid, the daughter cells are going to be haploid. Mitosis occurs in somatic cells or cells of the body, while meiosis occurs in germline cells that are found in reproductive organs. Mitosis occurs throughout the life cycle of an individual, while meiosis in human is completed after sexual maturity. In other words, if an individual doesn't reach at least the age of puberty, the sperm cells, the egg cells are not going to be produced normally. Mitosis is used for growth, repair, and asexual reproduction, while meiosis is used for sexual reproduction, which allows for genetic diversity to develop, right, and recombination of the genetic information, okay, which plays a role in genetic diversity. Now, let's take a look at a quick overview of meiosis. There are two stages. We have a diploid stage of the cells, and we have a haploid stage of the cells. Now, in this graphic representation, we have the diploid stage shown in this color, orange, while the blue stage is blue. The haploid stage, is blue. So specifically, you start with a germline cell here that is diploid. That germline cell is going to undergo meiosis 1. And we said earlier that meiosis 1 is called reduction division. So here, if you track, for instance, these two homologous chromosomes, they will each end up in different daughter cells. So this one end up in this cell right here, while this chromosome end up in this cell. Because a chromosome you receive from dad is now in a different cell from his homologous that you receive from mom, now we say that this cell actually is a haploid cell. Now, meiosis 2 is an equational division during which the two sister chromatids are going to separate. So now you see this sister chromatid right here and this sister chromatid right here. Same thing for this other chromosome where this sister chromatid comes right here and this sister chromatid comes right here. Same thing applies in this cell. At the end of the day, you have 4 daughter cells. 1, 2, 3, 4. And these 4 daughter cells have half the amount of genetic information that was found in the germline cell. Now, what happens during meiosis 1? During meiosis 1, first and foremost, before meiosis 1, the germline cell will undergo interphase where the genetic information is copied. So we have the G1 stage, the S phase, and the G2 phase. Then the cell enters meiosis 1. During meiosis 1, in the early prophase, what we are going to have, and that early prophase is going to call prophase 1, right? So meiosis 1, prophase 1, metaphase 1, anaphase 1, and telophase 1. This is very important to remember, right? In the early prophase 1, we're going to have the homologous chromosomes that are going to sit side by side. The homologous chromosomes are going to sit side by side. We call this synapsis. Actually, you have a set of proteins that are going to force homologous chromosomes to to sit side by side. We call this set of protein the synaptonemal complex. I mean, I will not bug you with that. Okay? Now, so we go into see synapses. Homologous chromosomes line up the length of their body. And along with the synapses, we are going to have crossing over. A crossing over is when homologous chromosomes are going to exchange genetic information. Now, remember that you receive one set of chromosome from mom and one set of chromosome from dad. When we say homologous chromosome, we mean a chromosome you receive from mom is going to cross legs with a chromosome you receive from dad. Obviously, the chromosomes have to be homologous chromosomes. Very important to remember. In prophase 1 late phase, the chromosomes are going to condense. The chromosomes become visible, the spindle will start forming, the nucleus, the nuclear envelope will get disorganized, the spindles are going to target the kinetochores, and the chromosomes are going to prepare to meet on the equator line. And again, the spindle fibers here are the one that are going to cause this to happen, right? So then we move to metaphase 1. In metaphase 1, the homologous chromosomes sit on the equator line. Now, what is worth mentioning here, it's that the way this pairs of homologous chromosomes are going to line up on the equator line is completely random. So the way this set of homologous chromosomes are going to line up and this one is going to be independent, right? If we had a third pair represented here, we will also show that the way that third pair is going to line up will also be independent of the second pair or the first pair. So this is going to cause chances of genetic shuffling, what we call independent assortment. But that's another story. We're going to address that a little later. And then that metaphase 1 is going to be followed by anaphase 1. During anaphase 1, a set of chromosomes you receive from mom is going to migrate in one side, while the one you receive from dad migrate on the other side, right? And this is going to happen for all the homologous chromosomes. So the pairs are going to separate. Now we are in telophase 1. In telophase 1, there is going to be a quick reformation of the nuclear envelope. A cleavage furrow will develop, and the cell is going to snap in two. So now we've reached a point where homologous chromosomes are now in two separate cells. We are really in a division, a reduction division here. Okay. What else do I need to mention? Oh, cytokinesis is happening here because you need a distribution of the cytoplasm as well. Following that meiosis 1, we're going to have meiosis 2. So basically this slide, it's basically what I described earlier. Okay, prophase 1, early stage and late phase. Here we have crossing over. We have a description of what crossing over is going to be. Say, this is the duplicated chromosome you receive from dad and the duplicated chromosome you receive from mom. Notice that when you look the length of the chromosomes, you have at every single location what we call a locus, genes that are matching. We have A here, capital A. We have lowercase here. We have capital B here. We have lowercase b here. Now, what you are shown here is that these chromosomes are going to have the exact same genes, except that these genes may be in alternate forms, right? So we have one allele here, capital A, another allele here, capital lowercase b, okay? Lowercase a. Likewise, you have capital B here, one allele, and lowercase b here, a second allele. And you can do that for all the genes. They are at the same location, even though they are in alternate forms. Anyway, so in this set of chromosome you receive from dad, you have one sister chromatid and another sister chromatid. The one you receive from mom, you have one sister chromatid and another sister chromatid, right? So you have a crossing of the legs here, one from dad, one from mom. They cross here where we call a chiasma, okay? And eventually, this is the crossing over here. Eventually, when the crossing over is completed, we see an exchange of genetic information between the leg from mom and the leg from dad. So we have some of the dad segment here that is now in mom's leg, and some of the mom segment here that is now in dad's leg. Now, if we follow these chromosomes up to the very end of meiosis, then we are going to have that this chromosome here will end up in one gamete. This one here will end up in a different gamete. And this one here in one gamete. And this one here in a different gamete. Now, at the end of the day, when we look at these gametes, we're going to come to the conclusion that this gamete is genetically different from this gamete, which is genetically different from this gamete, which itself is genetically different from this gamete. Which means that at the end of the day, there are never two gametes that are the same. And this accounts for genetic diversity. So metaphase 1 is when we are going to see the law of independent assortment, meaning that the way these two homologous chromosomes are going to line up on the equator line is completely independent of the way these two are going to line up in the interphase. In metaphase 1. And understand that we have 23 pairs of chromosomes. So the recombination, not the recombination, I'm sorry. The assortments, you have 2 to the power 23 chances of assortment. That is a lot. That is a lot, okay? So this is the reason why we are going to have a lot of genetic diversity. This is over 8 million chances. 8 million chances, over 8 million chances. Or 8 million possibilities over that. Okay, so the law of independent assortment, you are shown here, okay, how you go from this diploid cell, right, this diploid cell to this haploid cells. So this is in metaphase 1. So metaphase 1, the chromosomes can align like this. In the same metaphase 1, chromosome could very well be aligned like this, which alignment is different, right? And then you will end up having gametes that are completely different here. Okay, so you have two possibilities in metaphase, and you have this in anaphase. After meiosis 1. So anaphase 1 and telophase 1, I already explained that, so I'm not going to spend more time here. And then meiosis 2. So now, between meiosis 1 and meiosis 2, there is no interface per se. Well, let me say, there is no DNA replication. There is no DNA replication, right? So the cells that the daughter cells in meiosis 1 are going to undergo another cell division, which is actually very similar to mitosis, in the sense that the mitotic spindle are going to now target the centromeres here, the kinetochores here and here, and they will start shrinking toward opposite direction. And by shrinking toward opposite direction, right, we're going to have metaphase 2. From metaphase 2, we're going to move to anaphase 2, where we have the two sister chromatids that are now going to separate and end up in different cells. Eventually, we have telophase 2, where we have nuclear envelope that are going to reform here. Okay, and you have a cleavage furrow for each cell, and eventually you're going to have 4 daughter cells that are haploid and genetically non-identical. So this is basically to reshow exactly what we covered, okay? The chromosomes are going to condense again in prophase 2, metaphase 2, they're going to line up. Anaphase 2, the sister chromatids are going to separate. And telophase 2 / cytokinesis, we have brand new daughter cells. At this point, we have 4. All right. So the result of meiosis is going to be 4 non-identical haploid daughter cells. Each is going to have a separate assortment of genes because of 1, we talked about independent assortment. 2, we talked about crossing overs. Now, because of the independent assortment, we have about 8,388,608 possibilities. This is without taking into account crossing overs. Now, if you add to that crossing overs, then you have limitless genetic diversity. That is the very reason why, even within a family, you cannot have 2 siblings that are genetically identical. It's not possible. All right? This is also the reason why it's easier now to solve crimes when we know how to use DNA. And that is the very reason why in forensic science, DNA is the go-to technology to use to solve crimes when we don't know who was present at the crime and who committed the crime. So at this point, let's take a look at how sperms are made. Well, sperms are going to be made in the seminiferous tubes. In the seminiferous tubes, you have that germline cells are going to differentiate to become what we call spermatogonium. A spermatogonium, it's actually a cell that is already determined to become a sperm cell. This spermatogonium is diploid. The spermatogonium is going to multiply by mitosis so that we will always have a batch of spermatogonium to go from. Then that spermatogonium is going to mature and become a primary spermatocyte. That primary spermatocyte is the one that can never go back anymore. That one is completely committed at this point to become a sperm cell. That cell is ready for meiosis. That primary spermatocyte will enter meiosis 1. After meiosis 1, we're going to have the production of two daughter cells. These daughter cells are going to be called secondary spermatocytes. Okay, this spermatocytes that are secondary are now going to undergo meiosis 2. And as they undergo meiosis 2, they produce spermatids. The spermatids are sperm cells that are not mature yet. A spermatid, even though it is a sperm cell, it is not a sperm cell that is able to fertilize an egg. That sperm cell still has to undergo maturation. And that maturation, it's a process, right? And all of that, it's basically to regulate reproduction, right? So through maturation, the spermatids that are haploid are going to become sperm cells, mature sperm cell. And the sperm cells are going to have a flagellum and a head. In that head, we're going to have a nucleus, and we're going to have a region, a little vesicle that contains enzymes that is called the acrosome. The vesicle is called the acrosome. Okay, so I did tell you that the sperm cells come from seminiferous seminiferous tubules. So you are going to have some diploid cells that are going to divide and match and partially differentiate to become primary spermatocytes. And this primary spermatocyte are going to undergo cell division and become secondary spermatocyte. And notice that there is some kind of envelope that keep actually the sperm cells connected as they are developed. Okay, so now we have a secondary spermatocyte. This secondary spermatocyte will undergo further cell division. So that further cell division is going to cause the production of immature sperm cells here. These immature sperm cells here are going to develop, okay, into this form that is now haploid, right? And after full maturation, we are going to have a fully developed sperm cell like this, 4 of them for every primary spermatocyte that undergo meiosis. So now, let's take a look at the details of the structure of a sperm cell. A sperm cell is going to have a head. That head is going to have a nucleus that contains DNA and a vesicle here that contains some enzymes. Okay? Now, to that head is going to be attached a middle piece here. That middle piece is going to be loaded with mitochondria. And these mitochondria are spiral mitochondria. And what is the job of mitochondria? The job of mitochondria is to produce ATP. We have a concentration of mitochondria here because the ATP produced by this mitochondria is going to fuel the movement of this structure that is the flagellum. And that flagellum is basically microtubules that are covered with a plasma membrane. So the microtubules are inside and the plasma membrane is on the surface. Okay? This flagellum is going to cause the sperm to move with a whip-like motion, right? Which is going to allow the sperm to travel after ejaculation. Now, let's address oogenesis. So we just got done with spermatogenesis, the making of sperms. Oogenesis is the making of eggs in females. So oogenesis is going to begin with oogonium. That oogonium is a diploid cell. This is a germline cell, obviously. In meiosis 1, primary oocytes are going to be produced, right? However, this primary oocyte, as they divide, are going to produce unequal size cells. You have one big cell and one teeny tiny cell. The teeny tiny cell is called polar body. Okay? Polar body. Now, that first polar body is going to further divide, but let me close that parenthesis. Then the big cell, the primary oocyte, is going to undergo another division to produce a secondary oocyte. Okay? So the primary oocyte divides unequally to forming a polar body and a secondary oocyte. Okay, so here we have a secondary oocyte and a polar body. In meiosis 2, this secondary oocyte is going to divide to form an egg, while an egg and a polar body, while this polar body is also going to divide to form some small teeny tiny cells. Now, we're going to have at the end of meiosis 2, 3 polar body cells that are good for nothing, and 1 huge, humongous egg cell. So, oogenesis is a process. However, a process that is discontinuous. Why do we say this? We say this because pre-birth, a female is going to first produce a batch of future egg cells. Then there is a stop until puberty, right? So you have a process that starts, then there is a stop, and then it's going to resume later. So oocytes are going to arrest in prophase 1 until puberty. So the batch of cells found in little girls are in prophase 1. Then when they reach puberty, the cells are going to go from prophase 1 to completing meiosis 1 to entering meiosis 2. So after puberty, meiosis 1 is going to continue and is going to halt at metaphase 2. And that metaphase 2, it's only going to be passed after fertilization. Mind that oogenesis is not completed without a sperm cell. Cells are stuck in metaphase 2 until these cells are fertilized by an egg. The egg is the one that is going to trigger the move from metaphase 2 to anaphase 2 to telophase 2. All right? Very important to remember. So a female is going to ovulate about 400 oocytes between the age of puberty and the age of menopause. However, most oocytes are going to break down. Now, let's take a look at a graphic representation of oogenesis. So we have an oogonium, which is a germline cell that is going to multiply by mitosis. That oogonium is going to mature and become a primary oocyte, which is a diploid cell. Now that primary oocyte is now the cell that can never go back to becoming oogonium, can only move on to becoming a secondary oocyte. After the first cell division, we have the production of a first polar body and a huge cell that we call the secondary oocyte. We are done with meiosis 1. And now, meiosis 2 is going to consist of the first polar body that is going to further divide into two polar bodies. And the secondary oocyte is going to divide into a large size cell that we call the egg and a teeny tiny cell that we call a polar body here. So that we have 1, 2, 3 polar bodies and one ovum or egg cell that eventually is going to mature and ready for fertilization. Now, during the course of what do you call that? During sexual intercourse, sperms are going to be released in the vagina, and they're going to travel through the uterine tube. Now, there are a number of factors that are going to help sperm cells to swim toward the egg. But let me close that parenthesis for a minute. The parenthesis of sperm cells. Let me close that for a minute. Let's go back to the egg. We have a primary oocyte. That primary oocyte is going to undergo meiosis 1. We have now a secondary oocyte. That secondary oocyte is going to be protected by a layer of cell, and that layer of cell is going to protect the secondary oocyte. That secondary oocyte, upon ovulation, is going to be released in the fallopian tube. This egg now, which is in metaphase 2, is going to travel through the uterine tube, encounter sperm cells, and be fertilized. Now, how does this fertilization occur, and how do we go from metaphase 2 to completing meiosis 2? So basically here, we are going to, well, actually, maybe before we dive into that, let's first of all compare oogenesis and spermatogenesis. So in the prenatal stage, oogenesis starts with the oogonium that undergo mitosis and producing primary oocyte. For spermatogenesis, we're going to have the production of spermatogonia. This spermatogonia actually will keep being produced in a young boy until the age of puberty. Now, let me say that the production of spermatogonia does not stop at the age of puberty. Okay, so I don't want to give you the false impression that it stops there. No, it doesn't. It it they are produced continuously, right? So the primary oocytes are going to be produced before birth in a female, and then they're going to be stuck at a certain stage. At puberty, though, the primary oocyte will start the meiosis process and produce secondary oocyte and again stop in metaphase 2, right? Now, that's for oocytes. While in spermatogenesis, we have the spermatogonium. At puberty, will start differentiating into primary spermatocytes. Meiosis 1 occurs, then we have secondary spermatocyte. And the process keeps going, and the future sperm cells stay connected in the form of spermatids. These are immature sperm cells. And eventually, these immature sperm cells are going to mature into sperm cells or spermatozoa. These spermatozoa now are going to come and interact with the secondary oocyte, causing a fertilization of the secondary oocyte, which is actually going to trigger the completion of meiosis 2 and the formation of a diploid cell that we now call a zygote. It's a zygote cell. Okay, that's a fertilized egg. So I still want to explain something here. Let me see. Okay, so let me show you that when a sperm cell comes, you have the flagellum that is going to push the sperm cell. The mitochondria right here are going to cause the production of the ATP needed for that sperm cell to swim. And the genetic information here in the nucleus, it's the only thing that is going to be released. The acrosome here is going to actually burst, and some enzymes here are going to cause a separation of this corona radiata, disorganization of this corona radiata, and the enzymes in the acrosome are going to digest the zone pellucida, which is made up of carbohydrate and so on, and see how the head of the chromosome, of the sperm, is coming to the membrane here. The head is going to release the nucleus in here. But before that nucleus is released, you are going to have anaphase that occurs and then telophase. So there are a lot of activities that are happening during fertilization that are happening concomitantly with cell division and eventually the merging of the two nuclei, the mother nucleus and the father's nucleus, so that we have a diploid cell that develops, and now we have one single nucleus. But let me go back a minute. Okay, so we talked about oogenesis and how oogenesis compares with spermatogenesis. Now, all of this doesn't always happen in, all of it is not always rosy and everything is good. Things unfortunately happen, bad things happen. Meiosis can be a process during which some alteration of the genetic information can happen. First and foremost, during the replication of DNA. During the replication of DNA during interphase, some spontaneous mutation can occur because of mistakes in the machinery of DNA replication, right? That's one. Two, during the whole process of meiosis itself, some mistakes can occur. For instance, one common mistake that can occur during the divisions themselves, it's what we call non-disjunction. That non-disjunction, it's a situation where the homologous chromosomes are not separating or the sister chromatids are not separating, which can cause some gametes to have more cells than other gametes. And the consequences can be, diverse. So this can happen in both males and females, right? Now, we know these days that there are some features that are more common in female as they grow old in age than in males, and also there are some features that are more common in males as they grow old in age than they would happen in female, which cause at the end of the day some children to be born with defects. We have the paternal age effect. That paternal age effect, it's basically that sperms are of less and less good quality as we age, right? And as we age, some genes become sensitive and become mutated. One particular gene that is mutated is the FGFR. FGFR stands for fibroblast growth factor receptor. That fibroblast growth factor receptor have been associated with a lot of birth defects in individuals because of the age of the father. And this FGFR gene actually gets mutated in the testes. Okay, so it causes all kind of issues. Now that's for the dad. In the mom's side, most of the time, what we do see, it's aneuploidy. Aneuploidy, it's basically when egg cells either have extra chromosome, we would call this trisomy, or that could lead to trisomy, or missing chromosome that will lead to monosomy, right? So what are some paternal age effects? There is a condition which is achondroplasia. Achondroplasia and all this paternal age effect are associated with the mutation in the gene FGFR, the fibroblast growth factor receptor, right? So achondroplasia, it's basically when the gene mutated is causing the formation of a short limbs and causing dwarfism to develop. The Crouzon syndrome, it's when the skull prematurely fuse. A baby is born with some fontanelles, right? Which means that the parietal bone are not merged completely with the frontal bone, right? While in individual with the Crouzon syndrome, this bone would already be merged, which is going to cause a problem, right? Which is going to cause a problem in terms of the development of the skull, right? So that individuals are going to have some bulging eyes, uh, beaked nose, just because of this malformation that are going to happen because of too early of a merging of bones. Hutchison Guilford progeria. This is basically when a child is getting old faster than the child is really, right? They're going to have a skin that is going to wrinkle very in a very short period of time. The hair is going to be very thin. The signs of aging are going to speed up tremendously in this individual just because of that gene that is affected. Multiple endocrine neoplasia. This is another situation that can lead to thyroid cancer, parathyroid cancer, and adrenal gland cancer. The Pfeiffer syndrome. This is another premature fusion of the bones in the skull. And the thanatophoric dysplasia. This is another type of dwarfism. Again, the bones are affected here. So this is where we are going to stop this first part of the chapter. Thank you for your attention. Bye now.